Hello everyone. Today we are going to see the topic enzyme inhibition, and it is the most important topic in biochemistry under the unit of enzymes. So before enter into the enzyme inhibition, we want to know what is enzymes. So enzymes are the biological catalyst which mainly speeds up the chemical reaction in our human body. So without enzymes our organ system will fail functions. So in every part of the organ there is some of the enzymes which activates our metabolism and gives us the energy. So enzyme inhibition in the sense, so the inhibitor, a substance which mainly inhibit the activity of the enzyme. So this is the enzyme inhibition. So coming to the enzyme inhibition, and it is a process in which a molecule called inhibitor binds to an enzyme and decreases its enzyme activity. So this inhibitor prevents the enzyme from binding to substrate properly. So normal reaction is the enzyme combines with substrate together form enzyme-substrate complex and it finally releases the product. But in case of enzyme inhibitor, in the place where substrate get binds, the inhibitor go and bind instead of substrate and no chemical reaction occur, hence no product will be released there. So this reduces or completely stops the chemical activity of the enzyme, and this inhibitors controls overall enzyme activity in the human body. So you could see the catalyst, which is the speed up the chemical reaction, forms enzyme substrate complex, and in inhibition the inhibitor will bind instead of substrate place in the active site and no chemical reaction will happen and it stops the chemical action also. And coming to the importance of enzymes inhibition, some inhibitors act temporarily while others act permanently. So many drugs like penicillin and antibiotics work by inhibiting several enzymes. So you could see the example of penicillin, which is the antibiotic. It mainly inhibits the bacterial enzymes. So bacterial cell wall formation enzymes it mainly inhibits. So if suppose any disease has occurred, the doctor used to give the penicillin, which finally inhibits the bacterial enzymes required for their growth. So obviously AC inhibitor is also a very good example. This acetylcholine esterase enzyme inhibitor is mainly used to treat the high blood pressure and heart failure, and thus enzyme inhibition has several important medical applications. And enzyme inhibition has very important in medical applications. Coming to the type of enzyme inhibition, it is broadly classified into reversible inhibitors and irreversible inhibitors. So in irreversible inhibition, the inhibitor can be removed and it just replaces the enzyme activity and just restores the enzyme activity, and it mainly causes a temporary loss of enzyme activity. So the reversible inhibitor can be reversed back and it just restores the chemical activity. And this substance mainly forms the covalent, non-covalent interaction with the enzymes. Non-covalent interaction in the sense their bonds are all weak bonds. So if it is easy to separate the inhibitor from the enzyme and restore the enzyme activity, and it is a temporary loss of enzyme also. And coming to the irreversible inhibitor, the inhibitor cannot be removed and the enzyme activity cannot be restored. Instead, it is permanently inactivated. So once the inhibitor will bind, it cannot be reversed back and it is irreversible, and it causes the permanent loss of enzyme activity. And moreover, this irreversible inhibitor forms the covalent interaction with the enzyme. So covalent interaction is it is having the strong bonds. So the bond is very strong and tight and cannot be easily removed by the external forces. So these are the main types of enzyme inhibition, reversible and irreversible. And further, reversible is classified into three types like competitive inhibition, non-competitive inhibition and uncompetitive inhibition. So in the following video we will see one by one what is competitive and what is non-competitive and what is uncompetitive. So mainly the slight differences will vary from one inhibitor to another inhibitor.